In studying the development of birdsong, there have been a number of remarkable discoveries, a number of which actually have had huge ramifications in terms of human brain function. So, what we're going to do in this segment is to look at just a couple of the methods that researchers have used in studying birdsong development, and some of the what I think at least, are very interesting findings that have resulted from employing those methods. One approach is to disrupt auditory input, and that can be done by simply rearing birds in isolation, like individual soundproof chambers. So if you're doing this, they can certainly hear themselves, but they can't hear any other birds. If it's a young bird, they'll have no conspecific model to copy syllables from, or maybe an entire song from, depending on the species. So they're on their own. All they can do is hear their own sounds, but nobody else's. Or you can go a step further and remove the cochlea. Okay, so no cochlea and therefore no hearing. You are totally deafening the bird. So not only is there no conspecific model because they're deaf, but now they can't hear themselves either. There's no auditory feedback. So basically what we're asking here is, do you need a conspecific model? Maybe you're fine if you can only hear yourself. Or what if you can't even hear yourself, therefore nothing else, no conspecific model? What, are you going to be able to produce any normal vocalizations? Well, if you're a ringdove, you can deafen the bird and not affect their vocal output. As we see here, we have sonograms on the upper portion of the screen. Over on the left is the caw of excitement. Just to remind you what that sounds like, and notice the sonograms on the top portion of the screen and the bottom are identical. So the bird is intact on the bottom and deafened on the top, and deafening has no effect, meaning they don't have to hear themselves or a conspecific model, and likewise for the bow coo over on the right. And again, you're comparing the 3 deafened birds up on top and 2 intact birds on the bottom, and there's been no alteration in the ringdove's vocalization. So that is kind of unusual, but remember, these are not songs, they're calls, and that can often make a huge difference. Researchers have noticed an interesting evolutionary trend, or at least what appears to be an evolutionary trend, by using these 2 techniques of isolation rearing and deafening. So we're just looking at a continuum here of the degree of environmental dependency. How much do you rely on experience in terms of developing normal behavior? And in this case, the normal behavior would be vocal behavior. And by environmental dependency, I'm referring both to auditory feedback, meaning they can hear their own vocalizations, as well as actually being provided with a model, a tutor, a conspecific model. So do you need to hear yourself? Do you need to hear a conspecific model? So let's see some representative species that have actually been looked at and how this all maps out. So there are some that have to both hear themselves and a conspecific model. And 2 representatives there are the white-crowned sparrow and the chaffinch. If you let them hear themselves but don't let them hear a conspecific model, then they're going to produce abnormal song. They have to have both. Then there are some that only have to hear themselves. They don't need a conspecific model. And the song sparrow is a representative species of that. Well, and then finally, there are some that need neither auditory feedback nor a conspecific model, and we just saw the ringdove and the chicken as well. But again, these birds are ones that don't produce song. They produce relatively simple calls. So again, what some people have conjectured here, and based on pretty good evidence actually, it turns out that the birds at the top, the white-crowned sparrow, the chaffinch, and even to some extent the song sparrow, are more evolutionarily recent than chickens and ringdoves, who are more ancestral. And so it appears that there may be a trend to, in the course of evolution, such that birds need more environmental input in order to produce normal behaviors. Various kinds of experiences like self-stimulation, hearing yourself, a conspecific model, that kind of thing. Okay, well, another approach to studying birdsong is to actually disrupt vocal output and see what happens in terms of the kind of vocalizations that you get as a result of that. And a good example of that is our own work on embryonic devocalizations with domestic mallard ducklings, Peking ducklings, in the embryonic stage. So if we look at the duck embryo, notice they hatch on day 27. So on day 23, this is the tucking stage. They're moving their bill upward toward the airspace, the blunt end of the egg. And then they tent that inner shell membrane, okay? And then within the next day, they'll actually break through it. And as soon as they break through it, they start to vocalize. And they still have 2 days to go before they're going to hatch. They're going to pip the outer shell on the next day and then cut their way around and hatch a day after that. So they're providing themselves embryonically with an awful lot of auditory vocal experience before they've even hatched. Okay, what happens if you don't allow that to happen? What if you devocalize them by getting to the embryo before it breaks into the airspace on day 25? In other words, get to them on day 24. So that's exactly what we've done. So the way we go about doing this is somewhere, maybe on day 23 or thereabouts. Candle the eggs, and I've shown you a candler in another segment, and all it is a light source, and we just hold the egg against the light source, which enables us to see pretty much through the blunt end of the egg so that we can draw around and mark the airspace. And you can see that little bump right there. This is an embryo that is, in fact, tenting, but is not yet broken through. How do I know it's not broken through? Look, if you look at thousands of these, well, I haven't looked at thousands in this procedure. Hundreds, yes. If you look at hundreds of these, you get to know pretty quickly if they're tenting or if it's too late and they've already broken through, in which case there's no point in doing this procedure. So, if all is going well, you've already marked the egg, you just very gently cut around the airspace, get rid of the shell over the airspace to get access to the embryo. And here's the bill right there that's tenting the inner shell membrane. By the way, if you just paint the inner shell membrane with a cotton swab with water, it renders it transparent, which of course we need to do so that we can actually see the embryo down below. Its normal state is like this, and you can't really see much below. We also need to avoid cutting the membrane to avoid cutting any major blood vessels. That's important as well. So after we've done that, we just make a tiny slit in the inner shell membrane, pull the bill out to about chest level, because you'll recall from another segment that's where the vocal organ, the syrinx, is located. Just pull the feathers apart a little bit and apply a topical anesthetic, and then make a tiny, very tiny incision with a very tiny scissors used for small animal surgery, and then just inject a surgical glue. It's called collodion. It was developed obviously for humans, not for birds, but it's liquid. But after about 40 seconds of exposure to air, it solidifies. So by doing this, we're actually coating the internal tympaniform membranes of the duck embryo. And at this stage of life, by the way, it's pretty much only the internal tympaniform membranes that are responsible for these embryonic and hatchling vocalizations. We don't worry about the externals. So we're coating the internal tympaniform membranes with collodion. Collodion is sticking to the membranes, and then it solidifies. And guess what? Now the membranes can't vibrate because they are basically solidified from the collodion. The nice thing about this procedure is about 48 hours after hatching, the collodion starts to wear off and they start to vocalize normally. But for our experiments, we're typically done with them after about a day after hatching. So again, there's the syrinx. Recognize that from another segment. And also this from another segment. So you can see when the bird is vocalizing, these inner shell membranes are pulled inward, but now they're coated with collodion, and they can still be pulled inward a little bit, but they're not going to vibrate. And then here the bird is not vocalizing at all. They're just hanging loose. So the bird isn't ready to hatch yet, of course. And remember, we're doing this on day 24. So we put it in its own little plastic box, put the plastic box in an individual soundproof chamber. So, because we haven't done anything to the ears, right? They can hear everything. They just can't hear themselves because they've been devocalized. We've taken away that ability, but we've left the auditory system intact. You see, if we remove the cochlea, they wouldn't hear anything and we wouldn't be able to test them to any sounds. That's the whole point of doing this, to find out what sounds they're responsive to in terms of, variations of the alarm call or the assembly call or whatever experiments we happen to be doing. But then we put them in auditory isolation, and then when they hatch, then we're ready to test them 24 hours afterwards. And they look like that. So they're just fine, but they can't really say much. They can't say anything. So those are the two vocalizations that are made by our control sham operate subjects. In other words, ones that have been injected with physiological saline just to control for the effects of surgery. And they vocalize normally. And over on the right, we have our experimental subjects that have been injected with collodion. In other words, they have been devocalized, and all you're hearing from them is puffs of air, no tonal sounds whatsoever. And here's what these vocalizations sound like. First, the control subjects, the long, slow note. Actually, it's a call, not a note. And then the short, fast call. And this is all our devocal ducklings are capable of doing. It's like getting an obscene phone call from a duckling or whatever. But no, they can't vocalize. They're basically just breathing. They're trying to vocalize. That's the thing. But that's the only kind of sound that they can make. Okay, so let's move on and look at birds that actually sing, that make these more complex types of vocalizations. So what you can do here is denervate the hypoglossus nerve. It's called hypoglossal denervation. And you'll recall again from another segment that it's the hypoglossus nerves that actually control the muscles of the syrinx on the right and the left side. And the hypoglossal nerves actually come from the brain. They're cranial nerves is basically what they are. Okay, so what you're going to do is cut one or the other. Which is really all you need to do, and you'll see the result of doing that in a moment. So here's normal chaffinch song. Okay, it looks like this, it sounds like this. Here is the result of cutting the left hypoglossus nerve. Oh my gosh, the song is a mess. It sounds like this. So that nerve obviously plays a huge role in terms of producing normal song. And it's coming from the left side of the brain. You'll see why that's important in just a moment. However, you only get this devastating effect on song production if you've cut the nerve after song crystallization. If you do this during plastic song, they'll produce normal song. Oh, and by the way, if you cut the right hypoglossus nerve, notice there's no effect. It's a perfectly normal song. So that's one approach. Now you can go into the brain and lesion nuclei, specifically HVC, probably the main song production nucleus. It's called, remember, it stands for higher vocal center from another segment. So here, there's been no lesions taking place, and you get normal song. What if you lesion HVC on the left side of the brain? Now, you're not taking away the left hemisphere. I'm just representing that here so as if to represent that we've messed up HVC on the left side of the brain. All that's missing is HVC on the left side of the brain. So you get that same disruptive effect, just like if you cut the left hypoglossus. What happens if instead you lesion HVC in the right side of the brain? You get no effect, normal song. Again, you get a similar finding. You only get that abnormal song if you lesion HVC on the left side of the brain after song crystallization. If you do it before, then it's going to produce normal song. But there's an exception here that we don't see with lesioning the hypoglossus nerve. The song will return to normal the second summer. It's only going to be abnormal in the first summer, their first summer of adulthood, when they would be trying to sing territorial and courtship song. Everything is remedied in the second summer. And that's what led to a really exciting finding regarding brain plasticity. This is actually the first evidence of the growth of new neurons after reaching adulthood. People used to believe that no, after adulthood, no new neurons. No, you're out of luck. No, not at all. And of course, we've now known differently since at least the 1970s when this research was first done. And of course, it generalizes to all other species, including our own. So that was a really exciting finding. And it kind of explains why you get this effect, because if you look at HVC's volume, meaning the number of neurons basically in HVC, the volume is quite large in the spring. And of course, that's when they're doing most of this singing, territorial song, courtship song. But they're not doing that singing in the fall and winter. And if you look at HVC's volume, it actually shrinks to a smaller size. There's a loss of neurons because the bird doesn't need all those neurons. It's not doing singing. It's doing simple calls, perhaps. Plastic song, yes, but not full adult song. So look at what's going on here. If we plot the volume of HVC along this timeline. So in subsong, volume is very small. And then, so that's right after hatching, basically. But then as they're getting older, okay, they're going to start engaging in plastic song. And again, we're looking at HVC here, the volume of HVC. And during plastic song, it continues to grow and grow and grow. After song crystallization, right there, it's very high. Very large volume to HVC, because now they're singing full adult song. But then, during the fall and winter, it's going to shrink back because they're singing plastic song again. That's what PS down here is abbreviated for. Not enough space to put plastic song. But then the second summer, FS, full song, it goes right back up to a large volume. And the reason for that is that new neurons have now grown in HVC. So remember, it shrunk back in the fall and the winter, and now new neurons will actually grow in HVC. So even though you lesioned it originally, it's only going to affect the first summer when you're destroying those neurons. But the new neurons are going to grow, and in the second summer, you're going to get normal song. So why doesn't it happen with the hypoglossus nerve? Because that nerve does not regenerate. If it did, then you would be able to see a similar effect. But once that you cut the hypoglossus nerve on the left side, you're going to end up with nothing but abnormal song forever and ever and ever, as long as you do it after song crystallization. So yeah, there's a great deal of brain plasticity. And again, HVC is what we're looking at here. So some pretty exciting findings that have led people down all kinds of interesting avenues of research in neuroscience in terms of looking at the relationship between the brain and behavior.